How would you define a chromosome? What do you picture in your head? In eukaryotes, a chromosome consists of a single molecule of DNA associated with proteins that help maintain its structure and function. You've probably seen a chromosome depicted like this. However, this commonly used image can be misleading, as it actually depicts a linked pair of identical sister chromatids after DNA replication. A single condensed chromosome actually should be depicted like this. This is one of several instances where imperfect terminology can make biological processes seem more puzzling than they actually are. So, in this video, we will give an overview of a few important terms and concepts that are used throughout biology. Every human cell has a mass of DNA associated with other proteins and RNA stored in the nucleus. We call this complex of macromolecules chromatin. You may have heard chromatin described as a tangled mess of DNA and protein, but we're starting to figure out that it does actually have its own organization. Each chromosome contains a single molecule of DNA associated with proteins to help it maintain its structure and function. Chromosomes can be uncondensed or condensed. The compact structure of a condensed chromosome allows the cell to more easily move and manipulate chromosomes independently, which is useful for cellular processes like mitosis and cytokinesis. Humans have 23 unique chromosomes, each including the proteins associated with a single molecule of DNA containing many genes, regulatory elements, and non-coding DNA. Biologists quantify this number of unique chromosomes in an organism, also known as the number of chromosomes in a haploid cell, such as ovum or sperm in humans, with the variable n. Therefore, we say that human cells have n equals 23. N changes between species and does not indicate how complex the organism is. For example, the cells of budding yeast, Saccharomyces cerevisiae, have n equals 16. The cells of fruit flies, Drosophila melanogaster, have n equals 4, while the cells of domestic dogs, Canis familiaris, have n equals 39. Even though humans have 23 unique chromosomes in each haploid cell, our somatic, non-sex cells are diploid. So, in most of our cells, we have two slightly different sets of these unique chromosomes. Therefore, we describe diploid cells as 2n, or as having 46 chromosomes in total. You receive one set of 23 chromosomes from your mom and one set of 23 chromosomes from your dad. These chromosomes contain very similar sequences of DNA, but have different versions of the same gene located in the same spot, or locus, on the DNA. We call these different versions of the same genes alleles. Also, because these chromosomes are very similar but don't contain identical DNA sequences, we call them homologous chromosomes. Same, homo, relation, logos. In sum, there are two types of cells in humans and many eukaryotes, haploid and diploid. Haploid cells have one unique copy of each chromosome, for example, n equals 23 in humans. Diploid cells have two copies of homologous chromosomes, for example, 2n equals 46 in humans. So what happens to our DNA and chromosomes when we have to make more cells through cell division? The series of biological events that occur during each cell division is called the cell cycle. In this video, we will focus on the mitotic cell cycle, with cells that undergo mitosis to create two daughter cells. When a human cell undergoes replication, every single one of our 46 homologous chromosomes is duplicated to form identical pairs. Each member in an identical pair of chromosomes is called a sister chromatid. Each sister chromatid is just a copy of one of 46 homologous chromosomes, and they are linked together into one unit. This is where you may have heard some people describe the cell as having 4n chromosomes, which can be misleading. These cells are still diploid with 2n or 46 homologous chromosomes, even though they are currently duplicated as pairs of sister chromatids. You didn't receive two more sets of new homologous chromosomes with different alleles, and the amount of unique chromosomes has not increased. Instead, you can describe this cell as diploid with 2n chromosomes and 4C DNA content. The C value stands for constant, the genome size or amount of DNA that is characteristic of a particular haploid cell. As such, a haploid cell will have 1C DNA content, and a diploid cell that hasn't undergone DNA replication will have 2C DNA content. This specific terminology allows us to describe how the diploid cell that has undergone DNA replication still has only 2 sets of homologous chromosomes, 2N, but now contains 4C DNA content. That is, this cell contains twice the DNA content it once had, 2C, because each chromosome has been replicated to form a pair of sister chromatids. This DNA replication ensures that at the end of each cell cycle involving mitosis, the cell can split these pairs of sister chromatids apart to divide into 2 diploid daughter cells with 2N chromosomes and 2C DNA content. Hopefully now you can explain the difference between chromatin and chromosomes and can describe how the homologous chromosomes duplicate to form pairs of sister chromatids when the cellular DNA is replicated. These concepts and their vocabulary are tricky, but are very useful to understand thoroughly when talking or thinking about biology. Thanks for watching.